Welcome to the Advanced Concepts presentation number 5 by myself, Mariella Roper. This presentation will detail the production of ATP from one glucose molecule and where every ATP is formed. Cellular respiration is the process of generating ATP from glucose. ATP is the common currency in the body and is used as energy to drive all biological functions. The generation of ATP is crucial for organism survival, with in fact many deadly diseases and poisons directly affecting ATP production and often having fatal results. However, ATP production takes place over the entire course of cellular respiration, which takes place in 3 steps: glycolysis, pyruvate oxidation, and the citric acid cycle. The citric acid cycle is then the precursor for the electron transport chain, where the majority of ATP is produced. The first step of cellular respiration, glycolysis, generates 2 pyruvate molecules with 1 molecule of glucose. Pyruvate is then later used to generate acetyl-CoA. However, in glycolysis alone, both ATP and FADH2 is generated. In the first and third step, hexokinase and the phosphofructokinase 1 catalyze the reactions, respectively, and each use 1 ATP molecule. In the sixth and ninth step, however, 2 ATP molecules are produced each, with the sixth step utilizing phosphoglycerate kinase and the ninth step using pyruvate kinase. In both instances, 2 ADP molecules are phosphorylated into ATP. However, even though glycolysis produced 4 molecules of ATP, because it used 2 ATP, the net gain of ATP in glycolysis is only 2 ATP molecules. Additionally, 2 NADH molecules are produced, which then transfer their electrons to reduce FAD to FADH2, which later yields ATP molecules in the electron transport chain. This will be discussed a little bit later. The second step in the process is the generation of acetyl-CoA from pyruvate. Through the sequential action of 3 enzymes and 5 coenzymes, known as the pyruvate dehydrogenase complex, pyruvate is oxidized to form acetyl-CoA, a 2-carbon molecule that acts as the fuel for the citric acid cycle. During this oxidation, 2 molecules of NADH are produced, 1 from each pyruvate molecule. This takes place in the 4th step of pyruvate oxidation, as dihydrolipoamide has to be oxidized back to lipoamide in order to start the process over again. The reaction is catalyzed by E3, or the 3rd enzyme of the pyruvate dehydrogenase complex. These NADH molecules will be useful in the electron transport chain later, where they will be used to produce ATP. The citric acid cycle is the 3rd step in the process and generates mostly precursor molecules that will enter the electron transport chain in order to mass produce ATP molecules. 2 citric acid cycles occur for each molecule of glucose, since each of the 2 pyruvate molecules produced from the glucose each produce an acetyl-CoA molecule. So in the 2 cycles, 2 ATPs, 2 FADH2s, and 6 NADH molecules are produced. More specifically, the ATP molecule is produced from a GTP molecule from the Krebs cycle, and the GTP molecule transfers a phosphate group to an ADP molecule in order to form ATP. This happens between the succinyl-CoA and the succinate step of the citric acid cycle with the removal of the CoA group. By the end of the citric acid cycle, the net production of molecules is 3 ATP, 4 FADH2, and 8 NADH. Though this may seem like a small net gain of energy, the FADH2 and NADH molecules will enter the electron transport chain and serve as crucial elements to the mass production of ATP. In fact, the so-called conversion rates are often discussed when talking about FAD and NAD molecules, since each NADH molecule yields 2.5 ATP, and each FADH2 molecule yields 1.5 ATP. These conversions take place in the electron transport chain, which takes place after the citric acid cycle. In the electron transport chain, the conversions, as just mentioned, take place to produce mass amounts of ATP. From glycolysis, the two FADH molecules will generate 3 ATPs with a net yield of 5 ATPs from glycolysis. In pyruvate oxidation, the two NADH molecules generate 5 ATPs. In the citric acid cycle, the 6 NADH molecules generate 15 ATP and the 2 FADH molecules generate 3 ATP. Along with the 2 ATPs produced as is in the citric acid cycle, this yields a total amount of 20 ATP. Thus, the total ATP generated from one molecule of glucose equals 30 ATP.